Good day everyone, we are the group 6 from BSN 1-10 and we will be discussing the digestion of carbohydrates, proteins, and lipids as well as the digestive enzymes involved. Our report consists of the following contents. Here are the objectives of the experiment. At the end of the discussion, the learners will be able to Number 1, to define what digestion is. Number 2, to determine what material should be used in the laboratory experiment accordingly. Number 3, to be familiarized with the various enzymes in the digestive system. And lastly, number 4, to identify the different reactions of carbohydrates, proteins, and lipids in digestion. What is the digestive system? The digestive system consists of the digestive tract and other organs that aid the body in breaking down and absorbing food. It is a long twisted tube that begins in the mouth and travels through the esophagus, stomach, small intestine, large intestine, and anus. Let us recall the enzymes. The digestive enzymes that are involved in digestion are the following. Amylase. Amylase is important for digesting carbohydrates since it breaks down starches into sugars. Next is maltase. Maltase is responsible for breaking down maltose, which is a malt sugar, into glucose, which is a simple sugar. The body uses glucose for energy. Next is lactase. It is an enzyme that breaks down lactose, a sugar found in dairy products. It turns lactose into the simple sugars, glucose, and galactose. Next is lipase. Lipase is responsible for the breakdown of fats into fatty acids and glycerol, which is a simple sugar alcohol. Next is sucrase. Sucrase is secreted by the small intestine, where it breaks down sucrose, the sugar in table sugar, into fructose and glucose. Lastly is proteases. It is also called peptidases, proteolytic enzymes, or proteinases. These digestive enzymes break down proteins into amino acids. Proteases are enzymes that are synthesized in the stomach and pancreas. The most significant are pepsin, that is secreted by the stomach to break down proteins into peptides, or smaller groupings of amino acids. Next is trypsin, that forms when an enzyme secreted by the pancreas is activated by an enzyme in the small intestine. Next is chymotrypsin, that breaks down peptides into pre-amino acids that can be absorbed by intestinal wall. Next is carboxypeptidase A, that is secreted by the pancreas. It splits peptides into individual amino acids. Lastly is carboxypeptidase B, that is secreted by the pancreas. It breaks down basic amino acids.
So now we will be discussing the digestive tract. Mouth. Mouth is the beginning of the digestive tract. In fact, digestion starts before you even take a bite. Your salivary glands get active as you see and smell that pasta dish or warm bread. After you start eating, you chew your food into pieces that more easily digested. Your saliva mixes with the food to begin to break it down into a form your body can absorb and use. When you swallow, your tongue passes the food into your throat and into your esophagus. Saliva contains an enzyme called salivary amylase that begins the process of converting starches in the food into a disaccharide called maltose. Another enzyme, lipase, is produced by the cells in the tongue. It is a member of a class of enzymes that can break down triglycerides. Esophagus. It is a tubular organ connecting the mouth to the stomach. It receives the food from your mouth when you swallow. The chewed and softened food passes through the esophagus after being swallowed. The smooth muscles of the esophagus undergo a series of wave-like movements called peristalsis that push the food toward the stomach. The esophagus does not produce any digestive enzymes, but it does secrete large amount of mucus that act as a lubricant, ensuring the smooth transit of the food bolus. Stomach. A large part of digestion occurs in the stomach. The stomach is a hollow organ or container that secretes gastric digestive juices and holds food while it is being mixed with stomach enzymes. When the contents of the stomach are processed enough, they are released in the small intestine. Pepsin is secreted by the stomach to break down proteins into peptides, or smaller groupings of amino acids. Small intestine or small bowel is an organ in the gastrointestinal tract where most of the absorption of nutrients from food take place. It is made up of 3 segments: duodenum, jejunum, and ileum. The small intestine is a 22-foot long muscular tube that breaks down food using enzymes released by the pancreas and the bile from the liver. It lies between the stomach and large intestine and receives bile and pancreatic juice through the pancreatic duct to aid in digestion. Exocrine cells in the mucosa of the small intestine secrete mucus, peptidase, sucrase, maltase, lactase, lipase, and enterokinase. The pancreas plays a big role in digestion. It is located inside your belly, just behind your stomach. It is about the size of your hand. During digestion, your pancreas makes pancreatic juices called enzymes. These enzymes break down sugars, fats, proteins, and starches. Your pancreas also helps your digestive system by making hormones. The pancreas contains exocrine glands that produce enzymes important to digestion. These enzymes include trypsin and chymotrypsin to digest proteins, amylase for the digestion of carbohydrates, and lipase to break down fats. The liver has many functions, but its main job within the digestive system is to process the nutrients absorbed from the small intestine. Bile from the liver secreted into the small intestine also plays an important role in digesting fat and some vitamins. The liver is your body's chemical factory. It takes the raw materials absorbed by the intestine and makes all the various chemicals your body needs to function. The liver also detoxifies potentially harmful chemicals. It breaks down and secretes many drugs that can be toxic to your body. Bile is not an enzyme. Rather, bile helps digestive enzymes digest fat. Bile acts as an emulsifier, which means it helps to mix fat and water. Gallbladder stores and concentrates bile from the liver, and then it releases it into the duodenum in the small intestine to help absorb and digest fats. Gallbladder has no enzymes, but there is a presence of bile. Bile is a fluid that is made and released by the liver and is stored in the gallbladder. The colon is made up of cecum, the ascending colon, the transverse colon, the descending colon, and the sigmoid colon, which connects to the rectum. It is responsible for processing waste so that emptying your bowels is easy and convenient. It is a 6-foot-long muscular tube that connects the small intestine to the rectum. The colon has no digestive enzymes. The rectum is a straight 8-inch chamber that connects the colon to the anus. The rectum's job is to receive stool from the colon, let you know that there is a stool to be evacuated, and to hold the stool until evacuation happens. The sphincters relax and the rectum contracts, disposing its contents. If the contents cannot be disposed, the sphincters contracts and the rectum accommodates, so the sensation temporarily goes away. The anus is the last part of the digestive tract. It is a 2-inch long canal consisting of the pelvic floor muscles and the 2 anal sphincters, internal and external. The lining of the upper anus is able to detect rectal contents. It lets you know whether the contents are liquid, gas, or solid. We will now proceed to digestion. So what is digestion? It is the process of changing food into simple forms. This process is catalyzed by enzymes which are secreted by the glands found either within the different regions of the digestive tube or outside the tube, emptying their secretions into the tube through their respective ducts. So why is digestion important? Digestion is essential because your body needs nutrients from food and drink in order to function correctly and be healthy. Nutrients include proteins, lipids, carbs, vitamins, minerals, and water. Your digestive system breaks down nutrients into smaller pieces that your body can absorb and utilize for energy, growth, and cell repair.
Now that we have already recalled the various enzymes, let us now dive deep into the digestive enzymes. So here we have the digestive enzymes classification based on foodstuff acted upon. So first in line we have the amylolytic, which is the carbohydrate splitting enzymes. Salivary amylase or ptyalin from the salivary glands. Pancreatic amylase from the word itself is from the pancreas. Invertices or disaccharides from the goblet cells of the small intestine. So next we have the proteases, which is the protein splitting enzymes. It is secreted as zymogens or inactive enzymes. Pepsinogen from the gastric glands of the stomach. Trypsinogen and chymotrypsinogen from the pancreas. And lastly, peptidases from the goblet cells of the small intestine. Next is lipases, which is the fat-splitting enzymes. Gastric lipase from the stomach and pancreatic lipase from the pancreas. And last, but definitely not the least, is the nucleases, which is the nucleic acid splitting enzymes. Ribonuclease and deoxyribonuclease from the pancreas. So now let's proceed to the general materials and equipment needed for the experiment. In this slide, we have the test tube, pipette bulb, and the test tube rack. Here we have the pinch clamp, crucible tongue, and test tube holder. Here you can see the different flasks that are used in the experiment. First, we have the Florence flask, Retard flask, and Erlenmeyer flask. And lastly, we also used a Bunsen burner and an alcohol burner. One of the most significant materials in the experiment are the reagents. So here are the list of the reagents used in the experiment. The reagents and chemicals. First, we have the iodine solution, followed by the Benedict solution, 1% sucrose, 1% maltose, 1% lactose, 0.4% of hydrochloric acid, 1% sodium carbonate, copper sulfate, toluene, 0.05 normal sodium hydroxide, and phenolphthalein.
We will now proceed to the digestion of carbohydrates. I will discuss the complete digestion and enzymes responsible for the digestion of carbohydrates. Complete digestion and enzymes responsible for the digestion. Ptyalin acts on starch in the oral cavity, changing starch to maltose. Pancreatic amylase on starch in the small intestine, changing starch to maltose. Maltase acts on maltose, changing it to glucose, while lactase acts on lactose, changing it to glucose and galactose. And lastly, sucrase acts on sucrose, changing it to glucose and fructose. Summary of the steps in carbohydrates digestion. Digestion of carbohydrates is performed by several enzymes. Starch and glycogen are broken down into maltose and then into glucose by amylase and maltase. Sucrose and lactose are broken down into glucose and fructose, or glucose and galactose, by sucrase and lactase. The enzyme salivary amylase that is produced by salivary glands in the mouth acts on polysaccharides, turning it into disaccharides and oligosaccharides. The enzyme pancreatic amylase produced by the pancreas in the small intestine acts on polysaccharides, turning it into disaccharides and monosaccharides. While the enzymes oligosaccharides produced by the lining of the intestine brush border membrane in the small intestine acts on disaccharides and turning it into monosaccharides.
So, here's the experiment for the digestion of carbohydrates by salivary amylase. So, the materials and chemical used is salivary amylase, starch, water, tubes, clamp, and pipettes. The reagents is iodine solution and Benedict solution. Procedure 1. Place 5 ml of 1% starch solution in a test tube. 2. Warm in a water bath at 400 Celsius, maintaining the temperature throughout the experiment. 3. Add 1 ml of saliva to the mixture and mix thoroughly. 4. At a 1-minute interval, transfer 5 drops to 1 depression on the spot plate and 5 drops of Benedict solution. Continue the test until the starch solution no longer gives a color reaction with iodine. 5. Treat the remaining mixture in the test tube with 10 drops of Benedict solution. 6. Place the test tube in a water bath for 3 minutes. 7. Observe. So here's the video we prepared.
In this investigation, we'll use an enzyme to break apart complex starches into simple sugars.
We'll use an iodine solution to test for the presence of starches. Normally, this solution is this yellowish color, but when this solution mixes with starch, it chemically reacts, becoming a very dark purple or black color. This makes it evident that starch is present in the solution. Our iodine solution has been used to show that both of these test tubes contain starch. To one test tube, we will add an enzyme that will digest these starch molecules.
The enzyme we'll be using is called amylase. It's the same enzyme that's found in your saliva, and allows your body to start digesting starches as soon as you begin to chew your food.
We'll place both of our test tubes into a warm water bath. This will allow our enzyme to work much faster.
Iodine makes the starch solutions black, but once the starch has been digested by the enzymes, that black color will go away.
You can see here that after our enzyme was allowed to work, that's exactly what happened, and the color in that test tube went away.
Now that the enzyme has digested our starch, we'll test to see if it was broken apart into simple sugars. To do that, we'll use Benedict solution. Benedict solution tests for simple sugars. When sugars are placed into this solution and heated, a precipitate is formed. That precipitate provides evidence that simple sugars were present.
We'll take some of our digested starch and place this into our Benedict solution. This will allow us to test for the presence of sugars. Then we'll place our solution over a flame so that it can be heated and observe whether or not a precipitate is formed. It's very important that whenever you heat the contents of a test tube, you take the proper safety precautions. Here you can see the precipitate at the bottom of the test tube.
So there you have it. This lab investigation provides evidence that when starch, a complex carbohydrate, gets broken apart into smaller molecules, those smaller molecules are in fact simple sugars.
So here's the result. So when iodine solution combined with the starch, iodine solution is normally yellowish in color. However, when mixed with starch solution, it brings forth a chemical reaction which turns it into a dark purple or black color. So it means that the starch is present in iodine solution. When iodine solution combined with starch and enzyme, in this part, it shows that the enzyme amylase from saliva, which digested the starch in the solution, and that's why the dark color returned back to transparent color. And next is the enzyme plus digested starch plus Benedict solution. The Benedict solution together with the enzyme that digested the starch, when heated, produces precipitate, which proves that simple sugars were present in the solution. Digestion of carbohydrates by pancreatic amylase. Most carbohydrate digestion occurs in the small intestine, thanks to a suite of enzymes. Pancreatic amylase is secreted from the pancreas into the small intestine, and like salivary amylase, it breaks starch down to small oligosaccharides containing 3 to 10 glucose molecules, and maltose. Materials and chemicals used: pancreatic extract, starch, water, tubes, clamp, pipettes. Reagents: iodine solution and Benedict solution. Preparation for extraction of pancreatic enzyme. 1. Remove the fats from a pig's pancreas and grind them in a meat grinder. 2. Place the pancreatic tissue in a flask and add 100 ml of water and add 50 ml of 95% alcohol. 3. Shake well and allow to stand for 2 days. 4. Strain the alcoholic extract through cheesecloth. 5. Test the pH of the extract and neutralize it. Procedure for digestion of carbohydrates by pancreatic amylase experiment. 1. Place 5 ml of starch solution in 2 ml of pancreatic extract in a test tube. 2. Shake well and place in a water bath at 400 Celsius for 30 minutes. 3. Divide the mixture into 2 equal portions. 4. To the first test tube, add a drop of iodine solution, and to the second test tube, add 5 drops of Benedict solution. 5. Observe. So here's the results. Amylase is present in human saliva as well as pancreatic juice. Starch can be detected by the addition of a small amount of iodine solution. If the starch is present, the sample will turn dark blue or black once the iodine solution is added. If there is the absence of starch, then the sample should be a clear light brown color. The complete digestion of starch to free glucose should result in no dark blue or black color when iodine solution is added. Benedict's reagent detects simple sugar. If the solution remains light blue in color, then it is negative for sugar. Meanwhile, yellow, green, orange, or red color indicates that maltose is present and is positive for simple sugars.
Preparation of intestinal extract. First, scrape the mucous membrane of the washed duodenum and jejunum of a pig's intestine. Second is grind the scrapings with washed sand in a mortar, transfer it to a flask, and add 50 ml of 1% sodium chloride and 5 ml of toluene. Allow to stand for 2 days at room temperature. After 2 days, shake the mixture frequently, then strain the mixture with a cheesecloth and store the extract in a refrigerator. The materials and chemicals used in this experiment are test tubes, 1 ml of 1% sucrose solution, 1% lactose, 1% maltose, 1 ml of intestinal extract, and last is Benedict solution. First step in this procedure is place 1 ml of 1% sucrose solution in each of the 2 test tubes. In test tube 1, add 1 ml of intestinal extract, and in test tube 2, add 1 ml of previously boiled intestinal extract. After the second and third steps, place the 2 test tubes in a water bath maintained at 40 degrees Celsius for 30 minutes. The last step is add 5 of Benedict solution to the 2 test tubes and observe. Repeat the procedure using 1% lactose and 1% malt. For sucrose with intestinal extract, it is a positive result for Benedict's test as sucrose splits up into simple sugar. Glucose plus fructose by enzyme or sucrases found in the intestinal extract. For sucrose with boiled intestinal extract, it is a negative result for Benedict's test in boiling. It breaks down enzymes causing it not to work. For lactose with intestinal extract, it is a positive result for Benedict's test happens when lactose splits up into simple sugar, glucose plus galactose by enzymes or lactase found in the intestinal extract. For lactose with boiled intestinal extract, it is a negative result for Benedict's test is during boiling, where it breaks down enzymes causing it not to work. For maltose with intestinal extract, it is a positive result for Benedict's test is when maltose splits up into simple sugar, glucose by enzyme or maltase found in the intestinal extract. For maltose with boiled intestinal extract, it is a negative result for Benedict's test in boiling. It breaks down enzymes causing it not to work. As you can see in the results, all the reagents with boiled intestinal extract are negative, and the reagents with intestinal extract only are positive. It is important to consider the levels at which enzymes can catalyze their target reactions, and their ability to maintain their structure are highly dependent on temperature. Boiling and freezing can deactivate enzymes easily.
We will now proceed to the digestion of proteins. I will discuss the complete digestion and enzymes responsible for the digestion of proteins. Protein digestion is a multi-step process that begins in the stomach and continues to the intestine. Complete digestion and enzymes responsible for the digestion. Pepsinogen in the stomach activated to pepsin in the presence of hydrochloric acid. Pepsin acts on proteins, changing it to peptides. Trypsinogen activated to trypsin by enterokinase in the small intestine, and chymotrypsinogen activated to chymotrypsin by trypsin. Trypsin and chymotrypsin acts on the proteins to change these to peptides. Peptidases act on peptides, changing these to amino acids. Summary of the steps of protein digestion. The liver regulates distribution of an amino acid to the rest of the body. In the stomach, pepsin breaks down proteins into fragments called peptides. Protein digesting enzymes are secreted from the pancreas into the small intestine. Amino acids are absorbed from the small intestine into the bloodstream. In the small intestine, a variety of enzymes break large peptides into smaller peptides and then into individual amino acids. A small amount of dietary protein is lost in the feces. The enzyme pepsin produced by the stomach chief cells in the stomach acts on proteins, turning it into peptides. Enzymes trypsin, elastase, and chymotrypsin are produced by pancreas in the small intestine. It acts on proteins, turning it into peptides. The enzyme carbopeptidase that is produced by the pancreas in the small intestine acts on peptides, turning it into amino acids and peptides. Enzyme aminopeptidase and dipeptidase produced by the lining of the intestine acts on peptides, turning it into amino acids.
Action of gastric enzyme. Turn a pig's stomach inside out, wash it with water and strip off the mucous membrane. Then, mince the membrane and place it in a clean bottle and completely submerge it in glycerol. Then, stir frequently and allow to stand at room temperature for 2 days. And lastly, decant the glycerol portion into a small flask and set aside. The first experiment for the digestion of proteins is by gastric enzymes. The reagents used are the following: 0.4% of hydrochloric acid, 1% sodium carbonate, copper sulfate, and toluene. For the experiment procedure, the first step is to place the following chemicals in 4 separate test tubes. Test tube A is 5 ml of gastric extract. Test tube B is 5 ml of 0.4% hydrochloric acid. Test tube C are 5 ml of gastric extract plus 3 ml of 0.4% hydrochloric acid. Test tube D is 5 ml of gastric extract plus 3 ml of 1% sodium carbonate. The second step is to add equal slices of coagulated egg white to each test tube and place the test tubes in a water bath at 40 degrees Celsius for 2 hours. The third step is to add 4 drops of toluene to each test tube and store until the next laboratory period. Take note that toluene is added to prevent formation of molds and to inhibit bacterial action. The fourth step is to determine the extent of protein digestion by noting the size of the coagulated egg white. Let's now proceed to the video experiment of the protein digestion by gastric enzyme pepsin. Here is the table for the test tubes with their specification. The first step is to place in each test tube the coagulated egg white, which is the boiled egg white. In test tube 1, add 10 drops of water plus 5 ml of pepsin. In test tube 2, add 10 drops of 1 molar hydrochloric acid plus 5 ml of pepsin. In test tube 3, add 10 drops of 1 molar hydrochloric acid plus 5 ml of pepsin, then place the test tube on an ice pack. In test tube 4, add 10 drops of 1 molar hydrochloric acid plus 5 ml of water. In test tube 5, add 10 drops of 1 molar sodium hydroxide plus 5 ml of pepsin. For test tubes 1, 2, 4, and 5, place it on a water bath at 37 degrees Celsius, then let it cool. After cooling, here are the results of the experiment. As you can see, each test tube differs from each other. For test tube 1, you can observe that the size is still the same as it looked before it was placed in the test tube, a small chunky cubes. It also shows a cloudy mixture. Its results make sense since pepsin without the presence of hydrochloric acid cannot truly digest food. For test tube 2, you can observe that some small chunky cubes are reduced in size and show a clear mixture. The egg absorbed most of the solution and was broken down. The presence of hydrochloric acid and pepsin changes into its active form pepsinogen, which is the key enzyme involved in protein digestion in the stomach. For test tube 3, you can observe that no chunky cubes are seen. Coagulated egg white suspended on the bottom. Precipitation can be observed. It also shows a clear mixture. For test tube 4, you can observe that all coagulated egg whites were broken down and suspended on the bottom. Also shows a clear mixture. Hydrochloric acid serves to break down foods as opposed to truly digesting them, which is the task of pepsinogen. For test tube 5, you can observe that the coagulated egg whites are suspended on the bottom and look the same. It became a yellowish color. It is because of the sodium hydroxide. The fifth step is to place 1 ml of the supernatant fluid from each test tube in separate test tubes. Then, neutralize test tube B and C with solid sodium carbonate. The last step is that in all test tubes, add 1 ml of 1% sodium hydroxide and 2 drops of 1% copper sulfate. This allows us to test for the presence of protein. For the second part of the experiment, here's another video that will be presented to show the result of sodium hydroxide and copper sulfate when added to a protein, which is the egg white. As you can see, the result of the experiment indicates that when the mixture turns blue, it indicates the negative result and it concludes that proteins are absent. While when the mixture turns to purple or deep purple, it indicates the positive result and in which concludes that proteins are present. Thus, the results imply that only the coagulated egg white in test tube C, which contains gastric extract and hydrochloric acid, has the most protein digested. Take note that protein is broken down by the combination of enzyme pepsin and hydrochloric acid. Not either works alone. The next part of protein digestion is the digestion of proteins by pancreatic enzymes. The reagents used are the following: 0.4% hydrochloric acid and 1% sodium carbonate. Experiment procedure: the first step is to place the following chemicals in 4 separate test tubes. Test tube A is 5 ml of gastric extract. Test tube B are 5 ml of gastric extract plus 3 ml of 0.4% hydrochloric acid. Test tube C is 5 ml of 0.4% hydrochloric acid. And test tube D is 5 ml of gastric extract plus 3 ml of 1% sodium carbonate. The second step is to add equal slices of coagulated egg white in each test tube and place them in a water bath at 40 degrees Celsius for 1.5 hours. The third step is to add 3 drops of toluene to each test tube and set aside until next laboratory period. The last step is to add 1 ml of copper sulfate to all mixtures. The results of the experiment is presented. It shows that when the mixture turns blue, it indicates the negative result and it concludes that proteins are absent. While when the mixture turns to purple or deep purple, it indicates the positive result and in which concludes that proteins are present.
Digestion of proteins by intestinal enzyme. Materials and chemical use: tubes, test rack, pipettes, milk, 5 ml of water, 5 ml of intestinal extract plus 1 ml of 1% sodium carbonate, 5 ml of boiled intestinal extract plus 1 ml of 1% sodium carbonate, 5 ml of boiled intestinal extract plus 1 ml of 0.4% of hydrochloric acid, and copper sulfate. Reagents used: 0.4 of hydrochloric acid and 1% of sodium carbonate. Procedure: 1. Prepare 4 test tube as follows. Test tube A containing 5 ml of water. Test tube B containing 5 ml of intestinal extract plus 1 ml of 1% sodium carbonate. Test tube C containing 5 ml of boiled intestinal extract plus 1 ml of 1% sodium carbonate. Test tube D containing 5 ml of boiled intestinal extract plus 1 ml of 0.4 hydrochloric acid. Step 2. Add 1 ml of milk to each test tube and mix thoroughly. Step 3. Divide the contents of the 4 test tubes equally and set aside the remaining 4 test tubes as control tubes. Step 4. Place the test solutions in a water bath at 40 degrees Celsius for 1 hour. Step 5. Add 1 ml of copper sulfate for both incubated tubes and control tubes. Results: Test tube A containing 3 ml of milk plus 0.5 ml of water has no changes in the solution, which means proteins are not present. Test tube B containing 3 ml of milk plus 0.5 ml pepsin has a change in the solution in the form of coagulation, which means proteins are present. Test tube C containing 3 ml of milk plus 0.5 boiled pepsin has no changes in the solution, which means proteins are not present. Digestion of proteins by intestinal digestion by proteases. Materials and chemical use: tubes, test rack, pipettes, 5 ml of 1% peptone, 5 ml of 1% peptone, 5 ml of 1% albumin, 5 ml of 1% casein solution, and copper sulfate. Reagents used: 1% of sodium carbonate. Procedure: Step 1. Prepare 4 test tube as follows. Test tube A containing 5 ml of 1% peptone, test tube B containing 5 ml of 1% peptone, test tube C containing 5 ml of 1% albumin, test tube D containing 5 ml of 1% casein solution. Step 2. Prepare another set of 4 test tubes containing the above solution and make these as the control tubes. Step 3. Add 3 drops of 1% sodium carbonate to 10 ml of the intestinal extract. Step 4. Add 2 ml of alkaline extract to test tube A, C, and D. Boil the remaining extract and add to test tube B. Step 5. Place all test tube in a water bath at 40 degrees Celsius for 1.5 hours. Step 6. Then add 1 ml of copper sulfate to all mixtures. Results: Test tube A in an incubated test tube and controlled test tube turns to be negative, which is blue in color. Test tube B in an incubated test tube and controlled test tube turns to be positive, which is semi-purple in color. Test tube C in an incubated test tube and controlled test tube turns to be negative, which is blue in color. And lastly, test tube D in an incubated test tube and controlled test tube turns to be negative, which is blue in color.
We will now proceed to the digestion of lipids. I will discuss the complete digestion and enzymes responsible for the digestion of lipids. Lipids are digested and absorbed in the small intestine. Gastric lipase in the stomach acts on emulsified fats, changing this to fatty acids and glycerol. On the other hand, bile in the small intestine emulsifies fats. Lipase acts on the emulsified fats, changing this to fatty acids and glycerol. Here are the steps of the complete digestion of lipids. Lipid digestion begins in the stomach with the aid of lingual lipase and gastric lipase. However, the bulk of lipid digestion occurs in the small intestine due to pancreatic lipase. When chyme enters the duodenum, the hormonal responses trigger the release of bile, which is produced in the liver and stored in the gallbladder. Bile aids in the digestion of lipids, primarily triglycerides by emulsification. Emulsification is a process in which large lipid globules are broken down into several small lipid globules. Then, lipases break down the lipids into fatty acids and glycerides. Fatty acids and monoglycerides are packaged into micelles that are absorbed by microvilli. Fatty acids and monoglycerides are converted back into triglycerides. The triglycerides aggregate with cholesterol, proteins, and phospholipids to form chylomicrons. The chylomicrons move into lymph capillary, which transports them to the rest of the body.
We will now proceed to the digestion of lipids. The materials and chemical used are the reagents used are Procedure. Step 1. Place the following in each of the 4 test tubes. Test tube A, containing 1 ml coconut oil plus 4 ml pancreatic extract plus 5 ml of water. Test tube B, 1 ml coconut oil plus 9 ml of water. Test tube C, 1 ml coconut oil plus 2 ml bile plus 7 ml of water. Test tube D, 1 ml coconut oil plus 2 ml bile plus 4 ml pancreatic extract plus 3 ml of water. Step number 2. Shake the test tubes well and place in a water bath at 40 degrees Celsius for 1.5 hours. Step number 3. Add a drop of phenolphthalein to each test tube. Step number 4. Add drop by drop of 0.5 sodium hydroxide using a syringe until the solution turns to a faint pink color. Step number 5. Record the amount of 0.5 sodium hydroxide used for each test tubes.
Here is the video experiment.
To test various substances on how they can digest lipid, we add a lipid cream with a pH indicator, which is pink at an alkaline pH, to each of these 4 tubes. To the first tube, we add water and bile salts. To the second tube, we add lipase enzymes from a pancreatic extract. To tube 3, we add lipase enzymes and bile salts. And to tube 4, we add another enzyme, amylase. And we simply observe these tubes over time, because if the lipid fat is digested, then it will produce fatty acids, which will then alter the pH of the solution and change the pH indicator from pink to yellow or white. And we look to see if there are any tubes where there is no color change, as in tubes 1 and 4, which make us conclude that water and bile salts do not digest lipid, and amylase, although it is an enzyme, it is specific to something other than lipid, and neither digest lipids into fatty acids. While there is digestion of the lipid triglycerides in both tubes 2 and 3 to the fatty acids, which change the color, this color change is far greater in tube 3. And so lipase works better in conjunction with bile salts. Bile salts emulsify lipids, breaking large droplets into smaller droplets so that the enzymes can access more lipid molecules as they catalyze chemical reactions.
Results. Test tube A containing lipid, water, and bile salt has no changes in the solution, which means lipid is not present. Test tube B containing lipid and lipase has a change in the color of the solution from dark pink to light pink, which means lipid is present. Test tube C containing lipid, lipase plus bile salt has a change in the solution, the color turning from pink to yellow, which means lipid is present. Test tube D, lipid plus amylase, has no changes in the solution, which means lipid is not present.
To sum up, carbohydrates are converted to glucose and monosaccharides, wherein proteins are converted to amino acids. And lastly, lipids are converted to fatty acids and glycerol. Therefore, these digestion products can be absorbed into the blood and transported to the body's cells.
We will now proceed to questions and answers. Question number 1. Give the effect of the following. A. Boiling the enzyme on its activity. B. Bile on fat digestion. C. Hydrochloric acid on gastric digestion. A. Boiling the enzyme on its activity. Boiling breaks down enzymes so they no longer work. The chemical bonds that hold together the structures of enzymes begin to break down. The resulting loss of three-dimensional structure causes enzymes to no longer fit their target substrate molecules, causing the enzymes to entirely stop functioning. B. Bile on fat digestion. Bile acts to emulsify fats, taking large droplets of fat and turning them into smaller particles. This increases the surface area that is exposed to that lipase, and the enzyme that digests fats can make the digestion process more efficient and faster. C. Hydrochloric acid helps break down, digest, and absorb nutrients such as protein. It converts protein to metaproteins, which can be easily digested. It also eliminates bacteria and viruses in the stomach, protecting the body from infection. It activates pepsinogen to pepsin. And lastly, it makes the pH of the stomach suitable for the action of pepsin.
Number 2. Proteolytic enzymes are secreted as proenzymes. What is the significance of this? So, proteolytic enzymes have many important functions in the body, including helping break down food for energy or on the skin, and are found in certain supplements. This might help digestion or with the breakdown of the proteins involved in swelling and pain. Suggests that they can improve digestion, decrease inflammation, ease arthritis, pain, and possibly reduce symptoms. Also, helps in regulating our metabolic functions and assisting in maintaining our energy levels and stamina.
Discuss briefly the activation of pepsinogen, trypsinogen, and chymotrypsinogen. Pepsinogen is an inactive zymogen and is secreted into gastric juice from both mucous cells and chief cells. Once secreted, pepsinogen will be activated by stomach acid into the active protease pepsin, and is largely responsible for the stomach's ability to initiate the digestion of proteins. Trypsinogen is generally regarded as the inactive precursor of trypsin and is activated by enteropeptidase. The pancreas stores inactive form trypsinogen because active trypsin would cause severe damage to the tissue of the pancreas. It activates all of the other pancreatic proteases, aphospholipase and colipase, which is necessary for the physiological action of pancreatic triglyceride lipase. Chymotrypsinogen can be activated by trypsin to form chymotrypsin, a digestive enzyme that breaks down proteins into smaller peptides. Chymotrypsinogen is a single polypeptide chain consisting of 245 amino acid residues. The rate of activation, maximum enzymatic activity, and the stability of the activity were found to be influenced by such factors such as pH level, temperature, trypsin concentration, cations, and inhibitor.
And for the last question, in these experiments on digestive enzymes, what factors influence the rate of enzyme catalyzed reaction? First is the temperature. Higher temperature results in higher kinetic energy, which causes an increase in the number of reactant collisions. Therefore, there is higher activity. Second is the presence of activators, wherein they bind to enzymes and enhance their activity. Third is the enzyme concentration. At a constant substrate concentration, enzyme activity increases with an increase in enzyme concentration. The greater the enzyme concentration, the greater the reaction rate. And last is the pH level. Enzymes will work best within the normal pH, and drastic changes in pH can result in the denaturation of the proteins. And that concludes our presentation. Thank you, Digestion, and we sincerely appreciate your attention.